So I'm very pleased today to introduce our speaker, Dr. Soo-In Lee from University of Washington. Professor Lee is the Paul G. Allen Professor of Computer Science at the University of Washington. She finished her PhD not too long ago at 2009 at Stanford University under Dr. Daphne Koller. Before she joined UW, she was visiting assistant professor in computational biology at the Carnegie Mellon University School of Computer Science and has received the National Science Foundation Career Award and has been named an American Cancer Society Research Scholar. Professor Lee is a leading AI researcher and computational biologist and is known for her pioneering work in developing and applying AI and ML to advanced biomedical science. She's widely recognized as a leader in explainable AI, which significantly enhances the usefulness of machine learning model interpretability. Her recent contributions to the biomedical field span two aspects. First, she has been applying explainable AI tools to biomedicine and is the creator of the groundbreaking SHAP framework that's now widely used to explain how AI models are arriving at the conclusions that they do. Second, she's applied this work to multiple biomedical research domains, spanning basic biology to clinical medicine. So I'm very, very pleased to have her here today, and I'll turn the podium over to her.
Okay, thank you very much. Thank you for the introduction. So today I'll be presenting our lab's research on explainable AI. It's a subfield of AI, and then I'm going to highlight its applications in a wide variety of problems, such as neurodegenerative studies and then various clinical applications. This talk is going to consist of two parts. So in the first part, I will give you high-level overview of many examples of what explainable AI can do to address new questions in understanding biological systems. And then, hopefully, I can convey the excitement of advancing this, AI and ML to make this possible. So in the second part, we are going to see how explainable AI can open new research directions in biology and healthcare, and that can give us a clue about how explainable AI needs to evolve and then improve to solve a wide range of real problems in biomedicine. So I'd like to start by thanking people behind the research I'm about to present, the Ames Lab. I'm leading the Ames Lab. And then, I work with these passionate researchers doing their PhD in computer science program or MD PhD program, pursuing computer science PhD. And then we work on a broad spectrum of problems, including developing explainable AI techniques, and then identifying cause and treatment of challenging diseases such as cancer and Alzheimer's disease, and developing or auditing clinical AI models. And then, again, with explainable AI as a core technology of our current research. So the key idea of explainable AI is to try to understand why a certain output was generated by a machine learning model. Say that, when you have a set of features from some individual and then a well-trained machine learning model, we know that we can predict various kinds of clinical outcomes. So such as, for example, imminent dementia onset, say, within the next 3 years. And then our, research, this is actual project published in JBHI a few years ago. And then our study seeks a tractable and then widely administrable set of metrics, so out of these features, and then they can accurately predict imminent dementia onset. So however, we know that in a traditional machine learning paradigm, we couldn't really answer a natural question of why a certain prediction was made. So it is because with large modern data sets, the best accuracy is often achieved by complex models, and then even experts struggle to interpret those models. So the idea of explainable AI is to explain the output of the black box machine learning model based on the contributions of individual input features for individual predictions. So this machine learning model then can represent a supervised or unsupervised task. It doesn't really matter whether it's a supervised model. And then my lab has developed a series of techniques. The first one being SHAP, SHAP, and it's a general framework that can be applied to any model, and then has some desirable, theoretical properties, which other methods in this domain do not. So just to show you, the basic idea of how it works, the key question is how much adding a particular feature I changes the model output, and that represents how much feature I is important. And then, doing that, so by doing that, this may, depend on which other features are already being used. Let's say a subset of features S, and then, the idea is to consider all subsets of features S, and then the average across them. So by doing that, this cooperative game theory guarantees us that the SHAP values or Shapley values satisfy a set of fairness axioms, which many other methods in this domain do not. And then it is indeed one of the most widely used methods these days. So, but it turned out that the developing SHAP did not solve the problem, and it actually opened so many research directions in this explainable AI field. So for example, in one work, we showed that SHAP unifies a large portion of explainable AI literatures with like 25 methods following the same principle. So all explained by removing features. And then this helps us to understand what goes wrong, understand the both failure mode and the success mode, and improve and develop new methods in this space. So on a computational side, we've explored many avenues to make SHAP computation even feasible and faster. For example, we developed a statistical estimation approach, and then approaches for specific models such as tree model, DIM model, and then most recently transformers. Okay, so now let's talk about how it is applied to biomedical problems. So using one of these techniques, for example, tree SHAP, the SHAP method for tree model, tree ensembles, and then we revealed individualized risk factors of chronic kidney disease or all-cause mortality or other clinical conditions. And then these results provided a new insight in terms of what matter, to those clinical conditions such as CKD. And then it can be applied to any kind of clinical outcomes. And when you have this X and Y, this method is going to tell you which X is important for individual patient. So in a different study, we also showed that the explainable prediction can improve a clinician's ability to predict events. For example, this graph here, shows the prediction performance for hypoxemia, low blood oxygen level, dangerous anesthesia complication. And then this ROC curve, it implies that clinicians can detect millions of additional hypoxemic events annually when they are aided by explainable AI. So quickly, just switching to another project to show you how explainable AI can be effective. In this project, we showed that explainable AI can help us interpret our biological age. So we named our method enable age, explainable biological age. And then here's how it works. For a 65-year-old person who turned out to be 72 in terms of a biological age, the explanations that increase or decrease the age come with the extent in the unit of years, and then which makes it super interpretable. So for example, cancer diagnosis is, of course, accelerates the biological age, and then age at cancer diagnosis or assistant C. And these explanations can also provided for various mortality causes. We also performed in our paper that's currently in press, Lancet Health and Longevity. We performed the GWAS study to identify potential genetic factors for age acceleration. So, which means biological age minus chronological age. So, speaking of specific cause of mortality, our lab performed several projects in the space of understanding the Alzheimer's disease, and it is one of the top 10 deadly diseases that has no cure, long-term effective treatment to reverse or slow down its progression. So critical barrier include the lack of biological understanding, molecular level understanding of the neuropathological hallmarks, whose building blocks are A beta and then tau. And then, so how can explainable AI help with that? So in the first project, I will show you the study published in Nature Communications a few years ago. So my student Nikasia Bebewang, she collected RNA-seq data sets from multiple cohorts. It's an all AD study. And then these studies have both of case and control tissues, brain tissues, and then at that point it was all bulk RNA-seq data, and you will see single cell data later. The idea is to use multitask deep neural network to describe the complex relationships between 20,000 gene expression levels with 6 neuropathological phenotypes. So in this problem, prediction is not, these are all post-mortem brain tissues. So prediction is not really the goal. The goal is to understand the complex relationships between X and Y, the genes, individual gene expression levels, and then these neuropathological phenotypes, which are complex phenotypes. So the advantages of using this kind of deep neural network is that we don't need to fully harmonize, synchronize the phenotypes across a cohort. So some of these cohorts, ACT or Mount Sinai study or Rosemap, each of them can miss some of the neuropathological phenotypes in this list. Still, you can do the model training. And then the major advantage is that you can capture complex relationships, the nonlinear relationship between gene expression levels and the phenotypes. So again, the question is why, in the mechanistic explanations of this complex relationship. So, and then, the XAI, explainable AI, helps, improves this sort of analysis in multiple ways. So the first is that it allows us to use this robust model, that trained across multiple cohorts, because we can later use the explainable AI techniques to explain the relationship between genes and the phenotypes. And then, for example, for one of the phenotypes, you can attribute the plaque count back to individual genes, and then that way you can identify the genes that are important for each of the neuropath phenotypes, and then get a sense of the genes that are universally important across many neuropathological phenotypes. So, and then in our study, that's Nature Communications a few years ago, we identified some previously unknown associations between immune response genes and then neuropathologies. And then later we applied a similar approach to cancer drug synergy prediction problem. So that's the most more recent paper, Nature Biomedical Engineering 2023. Okay, so now after we finished working on this, and we started really thinking further how to make the best use of explainable AI when you have a gene expression data set as input. So the current explainable AI attempts to explain the prediction based on the input features, but the individual genes are not as interpretable as biological pathways, some kind of functional unit. And then also the second improvement we made is the use of unsupervised modeling, which does not require the predictive labels for individual data sets. So the idea is that you want to reduce the dimensionality of gene expression data into lower dimensional space, embedding space, and then the decoder is gonna reconstruct the original data set, and then you don't want to lose much information, and then want to find the lower dimensional space. So that's how unsupervised deep neural network works, and then combining these two. Oh, and then in the explainable AI in that context, what it means is that you can identify genes that can explain the expression variation across the samples within the data set. So combining these two ideas, we designed, my student Joe Jenny Zach, he's an MSTP MD PhD student, and then he designed a new architecture that organizes individual genes into pathways. So you see in the bottleneck layer, in the embedding space, you use this pathway information, and so that each of the hidden variables means corresponds to each pathway, and then again you can attribute the reconstruction error to the pathways to get a sense of which pathways are important within that data set, and then in turn you can attribute back to the individual gene level. So this new method is more recently published in Genome Biology. And then the question is, what it means, if you apply this method to AD data set. First, we can extend the data sets because we don't have to worry about the phenotype data there. So we used an extended set of RNA-seq data set and applied this method. And then we identified the mitochondrial complex 1 as a potential mediator. So we first, identified the oxidative phosphorylation as an important pathway in this AD data set. Again, this data set, has, normal, the healthy control, and then it's a case control setting. So you see both types of, both kinds of brain data set. And then, this pathway was the most important to explain the expression variation across the samples. And then really cool thing is that, we were really lucky to work with Matt Caben's lab. And his lab has AD model, C. elegans model, expressing A beta. So we were able to do this in vivo test where we knock down this each gene in this mitochondrial complex 1, and then see whether there is any change in these AD phenotypes. Okay, so now then in a more recent study, we try to come up with a way to make an interpretable model for single cell data sets. And the single cell data sets are routinely collected to investigate the changes in cellular states between control cells and corresponding cells in a treatment condition, such as exposure to a drug or infection by a pathogen or a disease condition. So to better understand this, difference in the, treatment response, it would be great to, disentangle the latent space and then capture the variations uniquely enriched in these treated cells from those shared with controls. So this led to a new architecture to explicitly disentangle the data into shared and at the top, and then I'm not sure if you are seeing my arrow, and then salient, which represent the variation that are unique to the treatment condition. And it's a more recent paper in just published in Nature Methods. So when we applied this architecture to the data, data from drug-treated cancer cells versus untreated cells, we see that the cells behave very differently in the salient space depending on their TP53 gene mutational status. And then this was only possible because the shared space absorb all this cell type specific nuisance, cell type specific signals. And then we won't see this if we use just the conventional methods, because cell type specific signals are biologically more dominant signal compared to these treatment specific signals. So this kind of finding is extremely important for personalized medicine. And then TP53 is known to the patients respond differently depending on this mutational status to this drug. So we are continuing our research to understand the single cell level molecular signature underlying neurodegeneration, and then also treated versus control can also mean something like aged versus young, to understand biological aging process better. Okay, so in the second part of the talk, the rest of the talk, I want to show you the explainable AI. I want to show you that explainable AI is not just about interpreting the models and can even open new research directions in biomedicine. So the first example is a model auditing framework for clinical AI. And then in this study, we revealed that many published clinical AI systems to detect COVID-19 from chest X-rays learned shortcuts rather than genuine pathology. And then first, our results illustrated some, potentially dangerous situation where this algorithm appears to be highly accurate within one data set. That's the ROC curve, the red colored ROC curve. So it means that you did cross-validation test within one data set. And then still you are measuring the accuracy in the held out data set. And then if you use the data set that's external to the training data set, that's the result is the green colored curves. So it's a potentially dangerous condition that this algorithm does not generalize. So to understand why these models failed to generalize, we took a look at the saliency map. It's explainable AI method that's going to show you in the image which parts of the image were used by the machine learning model. For example, it's where the arrows are. So it's right that the model used a clear long basis as a way to determine that it's a negative COVID-19 status, but it's not really right that the model used the laterality marks on the X-ray image or medical devices the patient is wearing. It should not be used to predict negative COVID-19 status. So overall, throughout many explainable AI-based analysis, we basically stopped the field and moved to the wrong direction. There were already 6 published papers and hundreds of related models out there that learned this kind of a shortcut. So later, we sort of identified, the cause of this. So when the researchers created a training data set, they used mainly one source for the negative labels, and the newer database for the positive label. So you created like huge confounding factors when they created a training data set, and then that data set was used over and over by many different models. So and then we see that it's very important to dig into what an AI model is thinking. So the next target is dermatology AI models. in collaboration with Stanford Dermatology. And we found that, many cell phone apps, and then many prestigious journal publications that introduced these AI models. And then we showed the similar results. The results were consistent with what we saw in the COVID-19 project. They weren't really good when they were tested in the external data set. So when we try to interpret these models, and then we ran into an interesting challenge. So explainable AI as is not going to be usable here in this application. So say you have a dermoscopic image in the left, and then if you see, and then it's predicted to be benign, the current explainable AI method is going to show you a saliency map, and then the parts in the image are that are important are indicated with this red dot. So clearly, you need to go beyond just the feature attributions, which is basically the current state of the art XAI. So one idea is to create the counterfactual images. So the idea is that you generate the image, you modify your input image such that you cross the classification boundaries. So say that this image is this modified image, and then the prediction result will be malignant. So that way you will know what this AI model is thinking. So and the reasoning process, which is that when you see this at the center, this bluish region, it's called blue-white veil. And then it usually, indicates malignant. And then now you see that that's how, the AI model is reasoning. So, and then, in our preprint, currently in revision for journal, we developed a systematic framework to characterize this counterfactuals in collaboration with dermatologists. And then also another paper that can automatically label concepts from medical images. Okay, so now we also showed that explainable AI can help us to make the clinical AI more cost aware. So many clinical AI literatures we can find that they focus on getting prediction performance, better prediction performance, and then celebrate good accuracies, 99%, 95%. However, if you really think about how these clinical AI models are used, it's not, it's obvious that in some situations, gathering features is often costly and it's hard. So say that you have a patient with a traumatic injury in front of you, bleeding, and you want to predict whether this patient will have acute traumatic coagulopathy, ATC. It's time sensitive, it requires massive transfusion. And then, so earlier transfusion leads to better outcomes, less organ failure, or fewer, this post-hospital complications. So what that means is that fast diagnosis leads to fast treatment. So accurate prediction is important, but you really want to predict this fast. So in this case, time will be your cost. And then some of the features that need your deeper examination aren't the ones you want to rely on too much. So we developed this method called Co-AI, cost-aware AI. It is explainable AI based because explainable AI is going to tell you which features are important in general, and then we combine that information with the feature cost. We worked with EMTs and the medical directors and then our collaborator at UW, Dr. Nathan White, in emergency medicine department to get a sense of how much time it takes to obtain each kind of features. So and then the idea is to improve both the cost, decrease the cost, and then improve the accuracy. So our result is something like this. So you have in the x-axis, you have a model budget, and then within the budget, you can then measure the model's prediction performance, and then the co-AI method is better than many other alternatives, and one of them is the PEC score, that's the existing clinical score. And in terms of the accuracy, this PEC score is similar to co-AI when the model cost is much lower. So, and this approach is also applicable to other medical problems. So, and in our paper, we also showed that you applied to inpatient mortality prediction in ICUs, and we showed that co-AI improves accuracy of predictive risk predictions while lowering the number of features required to make them. And then we compared in this problem, there are many clinical scores already developed. So we show the co-AI is doing better. Okay, so in the last example, and my goal is to show you how explainable AI can help us open a new direction in the cancer treatment area. So the cutting edge in the treatment of cancers like blood cancer, acute myeloid leukemia, AML, it has increasingly become combination therapy. And then the rationale there is that by choosing drugs that target complementary biological pathways, we can achieve greater anti-cancer efficacy and fewer unwanted side effects. But then choosing the optimal combinations is hard. There are already hundreds of FDA-approved drugs, anti-cancer drugs. So then what that means is that there will be tens of thousands of possible pairwise combinations. So, and then also, different patients, even the patients who have the same type or subtype of cancer may respond differently to the exact same drugs because of the particular genomic characteristics. So when we have too many choices, it would be useful to know why or why not certain drug pairs are good options. And then, but more than that, in this project, we also aimed to understand the underlying principles of biological mechanisms by which two drugs have a synergy for a particular patient. So our method, we named it Express method. It's actually published. I forgot to update the slide. It's published in Nature BME 2023. It takes AML gene expression data and then consider for a pair of drugs. It also uses drug level drug pairs features such as the genes that are targeted by this, each of these drugs and the molecular structure. And then, you use high performance, complex model to predict drug synergy between this drug A and B. Not only that, it gives you explanations. So in terms of which genes are important and how much, and then you can then do the pathway level analysis to get a sense of which pathways are important, contribute highly to the synergy or the opposite of a synergy between these two drugs. So we found that the genes that are globally important, genes that are universally important for drug synergy, tend to be related to a hematopoietic pathway, which is linked to poor prognosis. And then we performed many analyses. All details are in the paper. We identified the underlying principles of a drug synergy. So the final conclusion was that stemness can be considered an axis to design combination therapies. What that means is that if you consider two drugs, you want to use one drug that's known to work well for less differentiated cancer, and then the other one being effective in more differentiated cancer. Okay, so I, first to show you, very brief idea, very briefly showed you that the explainable AI, even just feature attributions, it's a non-trivial problem. It's computationally very costly, and then needs, quite a lot of still, advancement from the ML perspectives. And then since the SHAP paper, we published many papers in this space, and then we are applying them to make meaningful impact in various problems related to first, medicine and healthcare, and then cancer biology and precision medicine, and also Alzheimer's disease biology. Okay, so just the briefly recap. I talked about many examples in terms of what explainable AI can do as is, feature attributions, and then the second part of the talk I try to give you a sense of how explainable AI can open expand and then open new areas, research directions in biology and healthcare. And then, just to mention a few next steps, and what, currently my lab is working on includes this expanding the explainable AI's focus, beyond feature attributions. So one is a concept-based explanations. The other is counterfactual explanations. And then also, important part is still feature attributions are important. And then, for more modern models, so like transformers or graph neural network, we need feasible solutions to compute accurate feature attributions. Also, the model auditing framework is really important when we see many, clinical AI devices, approved by FDA. You want to make sure that they are all, they are making the reasoning in the right way. So, and then, more, the story I was telling you today is only a subset of research we are doing. You can find more about our research on our website. And, one tip to, navigate, our publication list is that we work on many, different topics. And then I showed you this, A, B, and C, AI, and then basic biology and the clinical medicine. So we labeled every single paper in terms of which category is the closest, and also journal versus conferences, so you can find more about our research. Okay, that's it for today, and then I'm very happy to answer your questions.